In this video, we will talking about penicillin production. Penicillin is a type of antibiotic was first discovered by Alexander Fleming. In a simple word, we can define as antibiotic is a substance that produced by microorganism that inhibit the growth of other microorganism at its lowest concentration. Antibiotics simply term as secondary metabolite produced at late log phase or a stationary phase of their growth. Now we will look at the introductory part. Penicillin is a very important antibiotic used in the treatment of disease caused by gram-positive organism. Towards the gram-positive organism, it has a bactericidal activity. Penicillin are a beta-lactam antibiotic that inhibit the cell wall of gram-positive organism. Now look at the general structure of penicillin that contain thiazolidine ring, beta-lactam ring, acyl side chain. This much part that is known as a core region or a constant region that is known as a 6-APA, which is known as 6-amino penicillinic acid. It is a constant region for all kind of penicillin or a penicillin derivatives, and the acyl side chain that is only vary. Okay, if in the acyl side chain if it is phenylacetic acid, then we obtain the penicillin derivatives such as penicillin G, which is benzyl penicillin. If acyl side chain if it is hydroxyphenylacetic acid, then we obtain the penicillin derivatives that is penicillin X, which is hydroxybenzyl penicillin. Okay, this is how the structure of penicillin. The only vary in the structure that is acyl side chain. This much part that is always remain constant, which is 6-APA. Okay. Now we will see the steps involved in penicillin production. The first step is selection of microorganism. Then second step is selection of raw material. Third step is preparation of inoculum. Fourth step is fermentation process, and the fifth step that is recovery. We will see each and every step in detail. First step is selection of microorganisms. Mainly we use the fungi that is the Penicillium notatum and the Penicillium chrysogenum. Penicillium notatum has high efficiency for the production of antibiotic, whereas the Penicillium chrysogenum has long mycelia which use more amount of nutrient and grow more rapidly. So we transfer the gene of Penicillium notatum into the Penicillium chrysogenum. So ultimately we obtain the genetically engineered, which is Penicillium chrysogenum X1612. This strain we will use for the penicillin production. Second step is selection of raw material. While selecting the raw materials, we should keep certain thing in our mind, such as our raw material should provide an abundant growth for the mycelium, maximum accumulation of penicillin, and easy of extraction and purification of antibiotic. While selecting the raw material, it should not cause any kind of hindrance towards the purification or the recovery stage. So we have to take this certain points in our mind during the selection of raw material. As we know that the fungi does not grow frequently on the natural medium, it's required some synthetic or a artificial medium, such as carbon source containing glucose, sucrose, lactose, nitrogen source such as ammonium acetate, ammonium lactate, ammonium sulfate, trace elements such as Mg2 plus, Zn2 plus, which is required for the elongation of mycelia. Wheat flour is one of the main precursor element, which is the main source of phenylacetic acid, which convert to the penicillinic acid. Functional group from the pigment that add to the penicillinic acid, and the penicillin is produced. Third step is preparation of inoculum. In the preparation of inoculum, it is need to produce large amount of inoculum. Special design medium by Moyer and Coghill in 1946. This medium contain glycerol, can molasses, MgSO4, peptone, NaCl, CuSO4, FeSO4, agar and distilled water. There are the two types of method for the inoculum. is done by seed stage culture and the production stage culture. In the seed stage culture, we use the fungal spore, and for that we use the solid media, okay, whereas the production stage culture we use fungal mycelium and we use the liquid media, okay. Fungal mycelia is directly added into the fermenter and we mainly use the liquid media. There is the only difference between the seed stage culture and the production stage culture is that in the seed stage culture we use the fungal spore and we use the solid media, whereas in the production stage culture we use the fungal mycelia and in liquid media. There are the three methods for the inoculation is dry spore method, suspension of spore, and the third, that is the pellet inoculation. In the dry spore, the dry spores are inoculated in a fermenter with the help of microchapper. Microchapper is an instrument that will take up single spores and add into the fermenter, that is known as the dry spore method. Second, that is a suspension of spore. In the suspension of spore method, generally what happens, we prepare a suspension of spore in a distilled water or NaCl solutions which is used for the inoculation in the fermenter. Third, that is a pellet inoculation. In the pellet inoculation, suspension of germinated spore or mycelium which form a pellet at the surface of medium, so it is known as a pellet inoculation. Generally what happens, suppose this is our flask or a vessel in which the mycelia we inoculate, we add the spores of organisms, it will form the pellet at the upper layer, at the top of the layer. This pellet we directly add into the fermenter. Mostly the pellet inoculation is mainly used, okay, rather than the dry spore method and the suspension of spore. Fourth step is fermentation process. In the fermentation process, first of all we will take a fermenter. In the fermenter we will add the raw material or a synthetic medium in the fermenter. After adding the raw material we go for sterilization. After sterilization we will add the culture. Fungal mycelia is inoculated as an inoculum for the penicillin production. After adding the culture, we will maintain the temperature around 30 to 40 degrees Celsius. If there is an increase in the temperature, that will be maintained by the cooling coils or by the cooling jackets. pH we will maintain around neutral. Sometimes basic solutions such as NH4OH is used to maintain the pH. It will take around 72 hours to complete this process. And after 72 hours we go for the last step, that is our recovery process. In the recovery process, first of all we go for the filtration step. In the filtration steps we use the special type of filter. Special type of filter, here we use the rotary vacuum filter. Rotary vacuum filters are used that are situated in a completely sterile condition, because here we are dealing with the antibiotic penicillin, okay. So we avoid the chances of contamination, so we use the special type of filter to remove the fungal mycelia. Now what are the things which are remaining? That is two things. The first thing is a fermentation medium and the second, that is the penicillin that are present in the fermenter. Two things are that. For that we add the first of all, then we add phosphoric acid that will lower down the pH, that convert the penicillin to the anionic form, that will precipitate our forms. Then we immediately go for counter current extraction. In the counter current extractions we will add the organic solvent such as butyl acetate. After adding the butyl acetate, then we will back extract the penicillin into the water phase by adding NaOH or KOH. Then we obtain the salt of penicillin. Again we acidified by adding the phosphoric acid. Then again we extract with an organic solvent by adding butyl acetate, and again we back extract in the water by adding KOH or NaOH, and finally we get the penicillin salt. This is how in industry we produced penicillin.